Welcome, focused learners, to this explainer. I am absolutely thrilled to be your guide today. You can think of me as your resident cell biologist, here to take you on a truly fascinating microscopic tour. We're going to zoom way in, past what the unaided human eye can even see, to uncover the fundamental structural and functional building block of literally all life, the cell. Whether you're looking to ace an upcoming exam, or you just want a crystal clear understanding of what is happening inside your own body right at this very second, you are in exactly the right place. Let's get into it.
Here is our roadmap for today's session. First, life's boiling beginnings. Then, prokaryotic vs eukaryotic cells, followed by the great cell boundary. Next up, inside the cell factory. And finally, we'll wrap up with the ultimate cell study guide.
Section 1. Life's boiling beginnings, tracing the first cell. Let's step back in time, like way back to about 3.5 billion years ago. It's kind of the ultimate biological mystery, right? Where did the first cell actually come from? We know all living organisms are made of cells, but before the incredible diversity of plants, fish, birds, and humans even existed, life had to start somewhere. It's widely accepted by scientists that life originated in water, but maybe not in the vast open oceans. Some researchers actually believe life began in much smaller water pools with constantly changing, really extreme environmental conditions.
Well, to understand this, we don't actually need a time machine. We just need to look at extreme environments right here on Earth today. For instance, the hot springs of Puga Valley in Ladakh, India. Despite the freezing cold surrounding climate, these pools maintain temperatures near the boiling point of water. These blistering conditions pretty closely mirror what the early Earth looked like 3.5 billion years ago. And incredibly, life thrives here. The organisms living in these hot springs are mostly single-celled, heat-loving bacteria called thermophiles. By studying these ridiculously resilient survivors, scientists can model how ancient life first took hold.
Now, check out the physical chemistry of those early pools. Scientists from the Birbal Sahni Institute of Paleosciences in Lucknow studied these hot springs and found that calcium carbonate forms very rapidly around them. And these mineral deposits? Total game changers. They likely shielded early organic molecules from harsh radiation and extreme weather. But even more importantly, this calcium carbonate barrier may have helped form the very first protective membrane. And remember, without a membrane, there is no cell. It's the definitive boundary that literally separates life from everything else.
Section 2. Prokaryotic vs eukaryotic cells, the fundamental split in biology.
So, once the cell was established, life eventually branched out into two main structural categories. And this brilliantly illustrates our first category, a typical bacterial cell. Notice how all the genetic material is just sort of floating freely right there in the middle, in a region we call the nucleoid. There's no distinct enclosed vault holding the DNA. Plus, you won't see any complex membrane-bound internal structures here either. It's highly efficient for sure, but structurally very basic.
There's a massive divide between these two categories. Prokaryotic cells, like the bacteria we just saw, are incredibly tiny, usually just 1 to 10 micrometers in diameter. Fun fact, the word pro actually means primitive, and karyon refers to the nucleus. So they have a primitive nucleus with no membrane, and they totally lack membrane-bound organelles. In stark contrast, eukaryotic cells, the type that make up you, me, and the trees outside, are much larger, spanning 10 to 100 micrometers. They boast a well-defined nucleus, tightly enclosed by a membrane, along with specialized membrane-bound organelles. And while prokaryotes are almost always unicingular, eukaryotes can build massively complex, multicellular organisms, like us.
Section 3. The great cell boundary, walls, membranes, and the movement of water.
No cell lives in total isolation. They're constantly interacting with the fluid swirling around them. To understand how cells survive in their environments, we really have to understand osmosis. Every single cell has a plasma membrane that is selectively permeable, meaning it acts kind of like a microscopic bouncer at a club. It allows some substances through while completely blocking others. It blocks sugar and salt molecules, but it gives water the VIP pass. Osmosis is specifically the movement of water across this membrane. Water will naturally flow from an area where the water is highly concentrated, like a dilute solution with less solute, into an area where there's less water and more solute, until everything balances out nicely. But when we mess with that concentration gradient, the effects are honestly pretty dramatic. A balanced, healthy cell is perfectly stable. But if you drop a cell into a highly concentrated solution, like thick sugar water, suddenly there's way more water inside the cell than outside.
so water rapidly exits the cell through osmosis, causing the whole thing to dramatically shrink and shrivel up.
Okay, let's dive into this real-world experiment to see exactly why plant and animal boundaries differ so much. If you take a rayo leaf, which is a plant cell, and a human cheek cell, an animal cell, and you drop them both into a 20% sugar solution, osmosis kicks in for both. They both lose water. But under a microscope, the results look totally different. The animal cheek cell just shrivels up completely. Because it lacks a rigid wall, it has the awesome flexibility needed for an animal's moving tissues, but that makes it highly vulnerable to shrinking. The plant cell, on the other hand, has a secret weapon, an extra outer covering called a cell wall made of rigid cellulose. When the plant cell loses water, its inner contents shrink away, sure, but that stiff outer box remains perfectly intact. This is exactly why plants, which obviously can't run away from a drought or harsh weather, can still stand tall and withstand the elements.
Section 4. Inside the cell factory, a coordinated working system.
All right, let's move past the bouncer and step inside the complex, bustling world of the eukaryotic animal cell to see how it builds a complete living system. Everything inside that membrane is filled with a semi-fluid, jelly-like substance called the cytoplasm, and floating within the cytoplasm are subcellular components called organelles. Now, these aren't just random, floating blobs. They are highly specialized structures that you can only see under a powerful electron microscope. Working together, they build materials, take out the trash, and generate energy.
So a eukaryotic cell is literally a tiny living factory. Let's meet the different departments. First up, you have the CEO, the nucleus. Safely enclosed in a double-layered membrane, it holds your chromosomes and DNA. It's the master house of coded instructions directing every single cellular activity. Next, we have the manufacturing department, the endoplasmic reticulum, or ER for short. It's this vast spreading network. The rough ER is studded with ribosomes and acts as the protein assembly line, while the smooth ER is busy manufacturing fats and lipids. Once the products are made, they head straight to packaging and shipping, the Golgi apparatus. Acting as the cell's very own post office, it modifies, sorts, and packages these proteins into neat little vesicles. And finally, none of this runs without electricity, right? The mitochondria serves as the powerhouse, performing cellular respiration to churn out the energy that keeps this entire microscopic factory humming along.
Section 5, Ultimate Cell Study Guide, your master cheat sheet.
We've covered some incredible ground today, from boiling ancient springs to complex microscopic factories. Tell you what, go ahead and screenshot this slide right now. This is your master cheat sheet. We started with the cell itself, the fundamental building block of all life. We looked at thermophiles, those crazy heat-loving bacteria in extreme places like Puga Valley that give us a window into early life. We divided life into prokaryotes, primitive tiny cells lacking a defined nucleus, and eukaryotes, complex cells with prominent nuclei. We explored osmosis, the crucial diffusion of water across the bouncer-like cell membrane. We saw how the rigid cellulose-based cell wall protects plants, while animal cells rely solely on their flexible membranes. And finally, we toured the eukaryotic interior, visiting the nucleus, the CEO of genetic instructions, and the organelles, those incredible subcellular structures like the ER, Golgi, and mitochondria that literally act like specialized factory departments.
You know, it's truly awe-inspiring how much complexity exists in a space that is tinier than the head of a pin. We consist of trillions of these cells, all working together in absolute perfect harmony. But it leaves us with one final, pretty profound thought to ponder. If our cells are just tiny automated living factories, strictly following microscopic codes, who or what is really running the machine? Keep wondering, keep studying, and I'll see you in the next explainer.